Now, see, when you will take into consideration the microbiology, it includes the studies of microorganisms which are not seen with the help of naked eyes. Now, under this, there are different groups of microorganisms. One is bacteria, second one is yeast and molds, that is fungi. Third one is protozoa, rickettsia, algae, and the next one is viruses. Now, for what purpose these viruses have been studied? Viruses are the obligate parasitic organisms. Among these viruses, most of the viruses are pathogenic to human beings and animals. Now, when the morphology, cultivation, the reproduction of viruses to be studied, it will be possible to prevent these infecting agents, or it will be possible to eliminate that infecting agents by taking the specific precautions. By taking the suitable precautions, it will be possible to eliminate that microorganisms. Now, that is the aim and that is the objective of this subtopic, that is viruses. Viruses are obligate parasitic. Their morphological features will be studied, classification will be studied, and the pathogenic, the viruses which are pathogenic to human beings and animals, diseases were studied. Now, when you will take into consideration these virions, viruses are acellular. These are the biological entities which do not have the defined or definite cellular structure. Now, when you will take into consideration the prokaryotic organisms, vicious agents, they are too smaller in size. They are too smaller than bacteria. For their magnification, simple microscope or light microscope is not sufficient. To see the image of the virion, electron microscope is required. Now, when I was teaching unit number 1, I have told you about different types of microscopy. Different types of microscopy. Broadly, there are two main types of microscopy. They are on the basis of the source of specimen which is used for forming the image. In one of the microscopy, light rays are used. That microscopy is called light microscopy. In the second microscopy, instead of light rays, electron beams are used. There, that is called electron microscopy. Now, light microscopy has been further categorized into 4 different types, namely bright field, dark field, phase contrast, and fluorescence microscopy. Electron microscopy has been divided into 2 different types: transmission electron microscopy and scanning electron microscopy. Now, by using either of any one, that is transmission or scanning, the 3-dimensional picture of the virion has been obtained. The viruses are microscopic entities which consist of nucleic acid. Surrounded to the nucleic acid, protein coat, that is called capsid, is present. Now, these are capable of replication. These are capable to do replications when they will enter in the host cells. Now, when you will take into consideration the viruses based on the host in which the viruses will multiply. Viruses have been divided into 4 different types. They are, the first one is animal viruses are those viruses who have the capacity to infect animals and they multiply in the they multiply or they replicate in the animal cells. While replicating in animal cells, they will cause abnormalities in the animal cells. That type of virus is called animal virus. Now those viruses who have the capacity to infect plant cells and they multiply in plant cells and they cause abnormalities in plant cells, that viruses are called plant viruses. The third type of virula, that is the virus who have capacity to infect bacteria and they create abnormalities or they multiply in a bacterial cells. Such a viruses are called bacteriophages. Triangular facets and 12 vertices as well as 12 corners. Now imagine in front of your eyes the structure with 20 triangular facets. Matlab triangles, 20 triangles when joined together. The resulting structure will be formed, that is called exostrium, which is with 12 vertices and 12 corners. Now when you will take into consideration the general characteristics, these are the two different general characteristics. The third general characteristic is they are obligate parasitic organisms. Now the meaning of word obligate parasitic organisms are viruses are not having the capacity to grow in a culture medium. When you will take into consideration, the bacterial cell, which belongs to the prokaryotic group, you have studied that bacterial cells are, it is possible to cultivate bacterial cells in the laboratory. It is possible to cultivate bacterial cells in the laboratory. Now how the cultivation of bacterial cells is carried out? See, there are three essential components which are required for the living entities. They are number one, source of carbon, second one is source of nitrogen, third one is source of energy. Now when you will take into consideration these three sources which are given in simpler form or complex form, and they are specific ingredients are taken and they are mixed together and the medium is prepared. On that medium it will be possible to culture the bacteria in the laboratory. That media are called culture media. Here viruses are not at all cultured in the artificial culture media in the laboratory, but viruses for their cultivations always requires living host. Viruses for their cultivation always requires living host, and due to that reason this sentence has been framed, that is they are either of any one nucleic acid, that is DNA or RNA is present. In case of viruses, nucleic acids, DNA and RNA both never present, they are absent. Either anyone is present, both are absent. You have understood now? Either anyone is present. If you talk about both nucleic acids are absent. Both of the type of nucleic acids never seen in the virions. Now that nucleic acid which is present in the virion, that nucleic acid is used as a zoonotic material. The nucleic acids are either single stranded or double stranded. Now when we will take into consideration these 6 characteristics, see, on the basis of host, I already told there are 4 different types of viruses. They are animal viruses, plant viruses, bacteriophages and human viruses. When we will take into consideration the nucleic acids, on the basis of nucleic acid which is present in the virions, there are 4 different types of viruses. They are single-stranded DNA viruses which contains single-stranded DNA as a genetic material, viruses which contains double-stranded DNA as a genetic material, viruses which contain double-stranded RNA as a genetic material, and viruses which contains single-stranded RNA as a genetic material. Now, on the basis of nucleic acids present in the virions, there are 4 different classes of the virion. Broadly, there are 4 different classes of the virions. Now, when you will take into considerations bacterial cells, what bacterial cells will do? Bacterial cells will try to utilize nutrients. Bacterial cells will try to utilize nutrients. When cell wants to reproduce, the cell will utilize nutrients. The cell size will be little bit increased. Enlargement of the cell will take place. From both the sides, both the poles, the septum or cross wall will be formed. That septum or cross wall will be enlarged and it will be fused at the center. It will be permeable to some constituents during the preliminary stages. When in both the compartments equal distribution of components will be carried out, then that septum or cross wall will be completed. Virion in the form of the crop of the virion in millions of number will be released after lysis of specific host. Now, when you will take into consideration the general characteristic, these are 7 different points. When you will take into consideration another point that is called the morphology of the viruses, these are extracellular infectious particles. That extracellular infectious particles are called virions. They are much smaller than bacteria. They are the filtrable agents. They pass through the filter. They cannot have the capacity to hold like bacteria. When you will take into consideration these viruses, for their observations I told 2 times that electron microscope is required. Light microscope will not provide you sufficient magnifications. These viruses are called elementary bodies who have the capacity to infect human beings, animals and plants as well as bacteria. Now, in case of viruses, there are different symmetries with the head of the virion. The first and the most important symmetry is called helical symmetry. Now, when you will take into consideration the helical symmetry of the virion, there are different types of protomers. That protomers are the proteinic structures which are joined together to form the capsomers. They are bound together in the form of ribbon-like structures, and these structures are held together to form the helix. And this helix is present in the capsule. The characteristic properties of the protomers they have, because of which the helix will be held with the specific component, that is the nucleic acids. Now, when you will take into consideration these helical viruses, example of the helical viruses is tobacco mosaic virus, second one is rabies virus. Now, there are some viruses which are isohedral in symmetry or polyhedral in symmetry. Different types of protomers will aggregate together to form 5 or 6 forms of the capsomers, and these capsomers are joined together to form isohedrons. That isohedrons are with 20 triangular facets, 12 vertices and 12 corners. In case of isohedral viruses which are with identical cubical symmetry, there are 2 families. One is adenovirus and second one is poliovirus. Poliovirus is quite familiar. It is a positive agent of polio which is isohedral in shape or polyhedral in shape. In case of viruses, some of the viruses are surrounded by the envelope-like structure. that envelope is mainly composed of the phospholipids. You have studied under the bacteria topic fluid mosaic model of the cytoplasmic membrane. There you have studied कि phospholipid bilayer deflate is there. In that phospholipid bilayer deflate, proteins are buried. Some proteins are present towards periphery. Those proteins which are buried in a phospholipid bilayer deflate, they are called integral proteins. Those proteins which are present towards periphery, they are called peripheral proteins. Now here, the plasma membrane or cytoplasmic membrane, what components are present? Peripheral proteins, integral proteins and phospholipids. Out of that, with the interest to this topic, phospholipids are there. Similar type of phospholipids, saturated or unsaturated fatty acids, are present in the envelope of the virion, and that envelope of the virion is surrounding to the capsule. Now this envelope from the biocatalyst, different biocatalysts also, the viruses will be protected because of the envelope. When you will take into consideration the example of the viruses which are protected due to the presence of envelope, mainly herpes virus, hepatitis B virus and HIV virus. Now in case of fourth type of the symmetry which is seen in the viruses which are having the fourth type of symmetry, that is called a complex symmetry virus. Now when you will take into consideration this complex symmetry virus, these are the combinations of helical viruses and isohedral viruses. Now when you will take into consideration these viruses which are the combinations of helical and isohedral, they have two components in them, that is head and tail. Now when you will take into consideration head or tail, such a viruses are called complex viruses, namely bacteriophages are belonging to this group. Bacteriophages are those viruses who have the capacity to infect bacteria. Can anyone tell the example of the helical virus? It is listed in the right. Tobacco mosaic virus. Second one, example of the polyhedral virus. Adenovirus is a entire family of the virus or single virus? Is it entire family of the virus? Okay, thank you. Now when you will take into consideration spherical viruses, can anyone will tell me the example of spherical virus? Example of spherical virus. See, there are different types of complex viruses. That complex viruses are called bacteriophages. Amongst bacteriophages, will you enlist different bacteriophages, which I told before some time? Polyphages are the most extensively studied bacteriophages. Now, when you will take into consideration bacteriophages, there are, again I am repeating, T even phage and T odd phage. T even phages, T2, T4, T6, T8. Does I have told this? Yes. T odd phages are T1, T3, T5 and T7. Now, in addition, the other points related to the classification of viruses, that is primary characteristics, secondary characteristics, these things I will try to cover in the next lecture. I will stop here.